Hello, class. I hope you are doing great. So we are starting our chapter 3. So this chapter name is basically bioenergetics, enzymes, and metabolism. So before we start, there is a picture here, and you can, if you just quickly have a look at this picture, you can see bunch of fruits and vegetables on it, on this man. So basically, at this point, I want to ask a question. Do you like what sort of the diet you have? It? So if you quickly answer yourself, and what your friend likes. So maybe you are more vegetarian person, and your friend like more kind of like meaty products. So at this point, we can think about that different people have different eating habits and a variety of, they eat a variety of food. And some, as I mentioned, like vegetarian diet, some like the meat products, some are kind of omnivores. So it depends. Now, the question is, how is the chemical composition of our bodies held constant when our intake of food can be so variable? So basically, the answer lies in network of biochemical reactions, which is orchestrated by enzymes that convert one type of biochemical into another. So basically on the right side, you can see enzymes, and basically whatever we eat, we are, what our enzyme makes us. So no matter what, the end result for us is to get energy. So this is a Monday lecture. In this, we are going to talk about what is bioenergetics, thermodynamic and its laws, types of reactions, and free energy changes in chemical and metabolic reactions. And I will record the Thursday lecture totally different. I'll see how much I can cover. I just have a look at the time. Okay, let's start here. So bioenergetics. Bio means life. Energetics shows that it's come from energy. So it's basically a part of biochemistry, which concern with energy involved in making and breaking of chemical bonds in the molecule, which are found in a biological organism. So the definition wise, the study of the various type of energy transformation that occur in living organism is called bioenergetics. So now basically a cell need these energy transformation in order to fold macromolecules, produce waste, respire, movement, and we know lots of other things happening in this living organism. Now, we all know the energy is basically a capacity to do work or the capacity to change or move something. Now, a little bit high school, your science, thermodynamics, most probably you have done in your first year as well. And I will tell you why we are actually learning this thermodynamics, because these sort of laws are applicable to our, biological molecules. So thermodynamics is the study of changes in energy that accompany events in the universe. Now we will look a bit more in detail. So there are 4 laws that define thermodynamics. So we will cover all of them, and this will tell us in what direction an event will take place and whether we require energy to perform that reaction in our body or not. Plus, the other thing is that it never tells us that how fast this reaction or changes are occurring in body. So keep in mind, it's just guided us like in what direction the event is going to take place. And why it is important, it is really, really important to know when we are looking for a reaction in the cell. Like, let's say you are studying glycolysis, so we need to know like what would be the second product, and most probably the scientists need this information because you will see it's just an example, but most of like like reaction, if we know like we can target at or block that particular step of the reaction, and then that's what the medication and the new medicines come in. Now, so the first law of thermodynamics, we know that this also called the law of conservation of energy, and basically it states that energy can neither be created nor destroyed. We all know that. So then we talk about a little bit of transduction, which is the conversion of energy from one form to another. And basically, cells, they are also capable of energy transduction, where we can see, I will give you examples and you can see here in a minute. So basically, whatever the energy we have stored in our body, you can convert into some sort of another form of energy. And first of all, look at this. So in this, it's a mechanical energy windmill, and you can see it's converted into electrical energy, which is like outside the living organism or body. Battery, just to show you that store of electrical energy in a chemical form. Fan, it convert electrical energy to mechanical energy. These are not the living organism, or you can see inside the body these examples. What I meant to say is these with the fan, and these is just showing the conversion of one energy to another, but you can see these conversion form into animals or inside the organism. So look at this. It is kind of the same, in which a man is trying to actually trying to lift this weight here, and basically he is converting his chemical energy to mechanical energy by muscles. So we won't see like that he has some sort of a battery or something like that, but we know the chemical energy is in the form of food. He ate it and it's converting to mechanical energy, just like the fan, and through his muscles. Now, the other one example is the chemiluminescence, or basically you can bioluminescent organism. And most probably you heard these animals, they basically convert chemical energy to light energy. They glow in dark. And most probably the firefly, when you were young and very, very like, like you saw the children, they like to go and catch those fireflies because they are converting their chemical energy to light energy in dark. So those were example of transduction. Now, to understand more the first law of thermodynamics, we can divide the universe into system and surrounding. So what is system? So system is a subset of universe under study. For example, if you look at here, this is a flask, and by looking at this flask, this is your system, right? And surrounding are everything that is not part of the system. So we are not talking about whatever is in the flask. We are talking about whatever is outside this system is considered is your surrounding. So system and surrounding is usually we referred as universe. So a system energy changes can be now the change in heat content or performance of work. So now when we talk about a little bit more in detail here, any change, gain or loss of energy in the system must be accompanied by loss or gain in the surrounding. So if you see that there is a change, gain in energy, or you think that's a loss in energy in the system, it most probably by the surrounding. And the best example, you just made your tea, it was very hot, you took it outside in your backyard, it is a, let's say it's a fall or it's in winter, like mild winter, you know that the surrounding was cold, right? So you can see that the energy will escape and there is a loss of energy in the system, and you will feel that your cup of hot milk or your tea or coffee will like not any hot anymore in few minutes or so. So the energy, it means that what is the purpose of giving that example? The example shows that the system and surrounding, they have a kind of relation, and we need to consider both of them. And we are coming to this part in couple of few slides later on down the road. So now the energy of system is called the internal energy, and it's the changing during this transformation is called delta E. So most probably at this point you're thinking you started to teach us chemistry, but again in reality I'm not teaching chemistry. It's kind of a biochem kind of the course which I am going to merge, and we are actually incorporating these all sort of information, energy transduction, system, surrounding, heat into our molecules, into the living organism. So according to the first law of thermodynamics, we can, this is equation, you might have seen this, delta E equals to Q minus W. E is the internal energy, is the delta always represent the change, right? Q is the heat energy, W is the work energy. And depending on the process, the internal energy of a system, delta E, at the end can be larger, it's greater, it's equal to, or less than the initial internal energy. So it depends what sort of the process you were considering. Okay, so now let me go over a couple of more things. So we all know that if you do work, then definitely you will see that then there is a decrease in energy. And if you increase the heat, that definitely you are increasing energy. That's why we show it by science, which I'm going to show in a second. So in this system, first of all, before I go over this, this is an example here, right? So we will see this example here. So this is a system, and here we are showing you a picture. I try to put all the figures I am taking from your book, so you can go and you can read a little bit more in detail, because it's not, first of all, it's not possible. I don't like to write down, too much on the slides, because I don't like reading from the slides, to be very honest. And from in this, you can see I really like try to simplify the concepts. You will see it later, which you won't find it in book. So I'll show you, and then I think go over there. So this is a leaf of a plant, and during the day, what happens, the sunlight, of course, is being absorbed by the photosynthetic pigment, which is chlorophyll in the leaf, and it makes use carbon dioxide and make carbohydrate, right, like glucose molecule, which later on is incorporated and make the sugar molecules. So now here, so delta E is greater than 0, right? So if the cells absorbs light energy, its internal energy increases, and the energy present in the remainder of the universe has to decrease. But now here at night, what's happening, the energy relationship between the cell and its surrounding is reversed. And that's why I was saying in the previous slide, we need to consider our system with its surrounding because we have a direct relationship, right? So now what's happened at night between the cell and its surrounding, what happened, the carbohydrate produced during the day, these like source of energy, they are being oxidized and they are producing carbon dioxide in the mitochondria, and the energy is used to run the cell's nocturnal activities. And keep in mind, you have a lab, and these labs have been designed especially by keeping in mind what are the major, the chapters you are doing. You are isolating chloroplasts. You are looking whether those kind of like activities exist in that or not. So there are lots of like information you will get it when you perform those lab. So that's the reason behind it. Okay, now let's go back and we can see delta E of a reaction. We were talking about it either positive, either it could be negative, or zero if it's not definitely doing anything, right? But however, this alone cannot predict the likelihood of a particular transformation. So we can say it's all a balance between work done by or on the system and heat added or lost during a process. So if the reaction in a reaction that lose heat, they are called exothermic. Exo mean out, and if in during the reaction, if the heat is gained, that is called endothermic reaction, which I'm coming in a second to another point. So the second law of thermodynamics. So this is a like picture shown here, and here you can see a girl is actually holding a ball, and this rock, you can say that the ball falls off, from the top. Let's say she's dropping from a cliff to the ground below, right? So now, what do you think? What's going to be happen? So the additional work to move this ball has been reduced because we know we have a gravitational energy here, right? So now the second law of thermodynamic, if we look at a little bit and more, it says even in the universe tends to proceed from a state of higher energy to a state of lower energy. And once they do so, less energy is available to do work because, of course, some other stuff is going around them. So these events are called spontaneous because they can occur without the input of external energy. Keep in mind this example, and I'm coming in a minute to some other examples. Okay, so can we say at this point, this reaction is thermodynamically favorable, and you can say because it doesn't need any input of external energy. Okay, so along with every energy transformation, there is a loss of available energy due to tendency of randomness, which is called disorder, which increase whenever there is a transfer of energy take place. So I'll go in a second in more detail. What is the randomness of or the disorderness is? So let's see, we see that how, what's going to happen actually here. So look at this example of this statement, which I mentioned on the top. This is a giraffe, right? So it's eating peacefully, it's leaves, right, from the tree. So basically, not all food energy is available to animal having the meat because it's not possible. Some of the energy is actually like maintaining its like all cellular activities. Some of the energy in the leaf is going to go waste, right? So basically we can say that much of the chemical energy is the food is not available to the animal having that meal, and it same goes like with us. Let's say you go to McDonald's and you took a burger, which is kind of, I think maybe I don't know, like maybe like 680 690 calories in the burger, right, or something like that. So it is not but nothing 100% is available to everyone, right? Okay, so coming like giving you this information, do you think is there a machine that is 100% efficient? So most probably your answer would be no, and that's a reality. There is no such machine exist which can do 100%, which is 100% efficient. And we know that there are lots of, like either it could be a heat loss or anything associated with them, which is making it not 100% efficient machine. So now this second law of thermodynamics is bring us to a very, very important property, which is called entropy. So what is entropy? It's a measure of randomness or disorder, which I mentioned in the previous slide. So we can see in here, this is an example of, let's say, molecule show here. So it shows it's highly randomness. You can see molecules, like atoms are far apart. They have a high entropy, meaning that high disorderedness here. But when you compact these molecules, you can see the entropy is a little bit reduced. Here, when they are more compact, the low randomness, low entropy, low disorder. And I can give you an example, a very, very good example of a room that it's like, your room, if let's say somebody's room is messiness, there is a high randomness, like high disorder, chaos, high entropy. But when you just clean it up, organize your room, that low randomness and low entropy and low disorder. So now what happened here? Loss of available energy is equal to T delta S. So delta S is change in entropy between initial and final state. So you need to remember, I'm going to bring you an equation slowly and gradually. We talk about delta E first, right? And now we are talking about delta S here, which is called basically change in entropy. So keep in mind these little bit these terminologies. So now every event is accompanied by an increase in the entropy of the universe. So entropy associated with random movement of particles or matter, and it is energy not available to do additional work. So remember that point in under consideration. Keep in mind. Also, loss of available energy equal T delta S, and where delta S is change in entropy, which is kind of I explained. Let's see a little bit more in detail here. So basically, we can see here, this is an example. This is sucrose, let's say the cube, right? A one cube. And basically what you did, you dropped it in the, let's say, dissolve in water, right? So if I ask you whether the entropy is high or low when sucrose, like cube dissolve in water, so what's your answer? So you can see the randomness of this, the atom inside this sucrose cube. So basically there is a high entropy here, right? And same goes like when water molecule dissolve here and it starts converting to gas molecule, then we can say that entropy is high. So evaporation of water only increases the entropy because gases are free to move. That's another example. So events are accompanied by an increase in entropy of the universe. So these are just a couple of examples. We are not going in detail. I'm going to focus more on the biological stuff, but this is a base to go to there. So this is an example of a room I was giving you. So you can see here, this is a tidy room, right? If I ask which room has more entropy, so definitely your answer is the messy one. And we can see that entropy increase from, highly organized to disorganized. So this is a picture, so it will help you to remember the concept of entropy. I also I can ask you what's the symbol you represent, and I just mentioned here at the back slide that entropy, you are showing entropy showing by delta S, change in, right? Initial to final state. So this is the one we also done. So at this point, we can ask or like think starting relevant to our biology course. Is entropy high or low in nucleic acids and proteins? So what do you think about it? Pause the video, you can see, you can think about if your answer is low, so you are right. And the reason is because nucleic acids and proteins, they are highly compact, right? So there is low disorderedness, low randomness. What about energy level in nucleic acids and protein? So if we think about it, the more organized and the more compact they are, they have more energy. They have a high energy level. And if I ask you, can you explain it? Why? So basically, you are able to think about it and answer it, comprehend all those points because they are highly organized molecules and they have definitely a specific sequence, right? And at this point, I will again bring your attention to think about the organized room example. So maintaining a highly organized room, or molecule needs input of energy, and that basically comes and make them stay in the organized way. It's not that you came, let's say yesterday, you came from university late night and you just started putting your stuff, your bag at one corner and then shoes. So of course you are creating basically increasing the entropy, right? Because you don't have energy, so it shows the less level of energy, but keeping to highly organized room or these nucleic acid require input of energy, which you need to show by putting a little bit effort into it. Okay, so randomness is the opposite of directed work. So if you increase randomness, then you decrease the energy that can be used to do directed work. Now, I made these kind of you can see lots of these pictures and the slides just to explain the hard concepts. So what would be the entropy in a closed and open system? You know, there are three kinds of system. One is isolated system in which nothing can go in and out. Best example is a thermos flask, right? There is a closed system we can talk about. And let's take an example, swimming pool, right? Swimming pool is a closed system because the water, whatever the heat they are maintaining inside, it's not going outside, right? It's a closed system. Only one thing is coming inside. That's like human are jumping in. So it's a closed system, right? Then open system, you can say the best example is the human body because you know that various input like oxygen, food, water, they are our input, and the output would be like waste, carbon dioxide. These are our outputs. So, or even the matter exchange, we can say at this point. So the point I wrote here, available energy can never increase. So entropy, if you look at this point, the energy of entropy. So because energy is conserved, so you can say the entropy can never decrease because energy is conserved here. But here, entropy could pour out of the boundary, right? So entropy could, it decreases here. So keep in mind what happened in the open system, what happened in the closed system with the entropy. Basically, entropy can never decrease, right? Because available energy is not increasing at all, right? It can never increase. It's a closed system. Okay, so talking about this, this is the open closed system example. Let me think if I have to say anything about anything else. So yeah, that's I think pretty much about this. I will talk about a human example, I think, but I will talk in the later slide. Okay, now look at this. Living system maintain a state of order or low entropy. So now here we are talking about living system, and they maintain a state of order or low entropy. And here we must distinguish between system and its surrounding. So we talk about only the total entropy in the universe must increase. The disorder in one part, the system may decrease, but only if the surroundings disorder increases to a greater extent. So what I meant is, let's see this example again here. Can you explain whether entropy is high or low when sucrose cube dissolve in water, which we saw here? And explain whether entropy is high or low when water molecule dissolve, convert to gas molecule, which I was explaining in the previous slide. So we can say that because the water molecule convert to the gas molecule, then the entropy is high, right? Same because the disorderness is increasing, randomness is increasing. Now, bringing from that point into the cell here, this shows organisms decrease their own entropy by increasing entropy of their environment. And how they do it, look at this example. So this is amino acids are ordered into complex protein molecule. This shows the anabolism. One is a catabolism and the anabolism. So catabolism is the breakdown of the product. Anabolism is the creation of the product. And definitely regarding if we talk about this, so it keep in mind that the entropy plays a very, very vital role. And so organisms, they decrease their own entropy by performing these processes alternatively. Now, there is another concept which is called free energy. And basically, first and second law of thermodynamics indicates that the energy of the universe is constant, but the entropy continues to increase towards a maximum. So remember this example, always keep in mind here what's going to happen. Here, this is a water, this is the ice, right? And if you provide this heat or you put it outside, you can see that the entropy is increases to its maximum. Disorderness of the molecules, they are going to increase. Versus when water freezes, where entropy decreases because the water molecules of ice exist in a more ordered state with less freedom of movement than in the liquid state. So the decrease in entropy is particularly apparent in the formation of a snowflake versus the melting of this ice cube. So these concepts, like actually the free energy concepts, were combined by an American chemist, J. Willard Gibb, and it's expressed mathematically. This equation is called enthalpy delta G plus delta T delta S, or Gibbs energy. Delta G is written by enthalpy minus T delta, this here entropy. I know this is getting a little bit overwhelming, so I am going to the next slide and I'll show you how I organize. So just keep in mind this T is the absolute temperature, meaning you have to add 273 degree to get the Kelvin temperature. You know, we use Kelvin. Enthalpy is the total heat content, delta H, in a thermodynamic system. So if you want to get the thermodynamic total heat, you need to consider the energy, which is free energy, plus temperature with the enthalpy, and then you can get it. So to remember the Gibbs free energy, which is delta G equals to your enthalpy minus T delta S entropy. I kind of make a slide so it will make you to remember. So goldfish are horrible without tartar sauce. Look at this. This is a goldfish. They are horrible. The person is crying. This emoji without the tartar sauce. So what does it mean? Look, goldfish are horrible without a tartar sauce. So you can easily write down this equation, and if I ask, you can solve any problem, right? Now, this gives free energy. Basically, it could be less than 0, it could be greater than 0, or it could be equals to 0. So what does it mean? I make it more simplified, and I also mentioned you here regarding entropy, the process of how it basically increases and how it decreases when a snowflake forms. I will go bit more in another slide, but basically change in free energy is called delta G. It's a change in energy available to do work. Remember it, please remember this point. So entropy and free energy are two alternative means of assessing thermodynamic spontaneity. So basically, delta G is the measure of the spontaneity of a reaction. And if you plug in, let's say it's X, this is Y, and this is AB. And if you plug in this equation that the heat is 5 kilojoules per, and this is the temperature, and you solve this problem, the temperature and entropy, you end up with at the 10, you plug this, you get -5. Right, so this is the different values I put it. So when you get -5, it means the delta G is less than 0. When you get 5, it is delta G is basically greater than 0, and equals to 0 means delta G is of course 0. So that's what it meant here. So you plug in these equation, your numbers depend the heat you used in the system, the temperature, and the entropy produced in the system, which is you measure it and you plug these numbers in equation, you will end up with some value, and which is an indicator of your free energy. Again, the one important point from so far we done, entropy and free energy are the two alternative means of assessing the thermodynamic spontaneity. Okay, so basically, if I know this sometimes it's hard for students to remember, I'm like kind of like literally when I made these kind of slides, rather than just copy pasting, I didn't copy paste, I wrote those those things like so I made these kind of pictures. I like here, post this slide, so it will help you to understand. Again, this stick man is here, the ball is rolling toward him, and what can we see here? So we can see that less energy is required, right? And in the first few slides, I talked entropy is increasing, right? Disorderness of the system is increasing. At this point, can I say, what do you think? What would be the delta G here? It should be positive or it should be negative? So it is negative, it's less than 0, right? Versus this guy stickman who is taking the ball to the cliff, you can see, right? So he is actually using energy, right? It's greater than 0. It means it's plus G. So this is the few of the things which might can confuse you. But when you come back to the slide, like the disorderness, entropy increases, and I showed you the room slide and this, it will help you to get back to track. Okay, now look at this. So when it is less than 0, the reaction, because nobody is pushing this ball further down, here. So it's a spontaneous reaction, and we say decrease in free energy of system, increase in entropy of the universe. You can see, that's why I gave you an example of system and universe way in the first few slides, so you can get an idea what's going on. And this one, of course, it would be opposite. It is a non-spontaneous, or sometimes we call it spontaneous backward reaction. And of course, increase in free energy of the system, decrease in entropy of the universe, right? So this will always help you to get a concept of free energy. So this kind of reaction in which energy is you are releasing is called exergonic, remember it, and in which you required energy, right, to do something is called endergonic. So there are lots of terminologies they are going to mix up, but I hope these slides will help you to remember it. I also showed the same thing here, just in whatever the way you like and you can remember, please go ahead and remember. So all spontaneous energy transformation must have a negative delta G. That's what we saw, that stickman example. That is, the process must move to state to a lower free energy, and process with negative delta G are exergonic. Keep in mind this example. And process with positive delta G are endergonic. Keep in mind this example as well, right? Because we can easily see that it's release energy and it's required energy. Okay, so the magnitude of delta G indicate the maximum amount of energy that can be passed on for use in another process, but definitely not how rapidly the process will occur. So this is the key point. Please remember that thing in this will help you a lot. Okay, again, this is a summary slide, whatever you like. I just mentioned what are exergonic and endergonic reaction, which way, whatever you like the most, just keep in mind. The question is definitely coming from this slide too. Okay, so which reaction cannot occur spontaneously? Now, answer, please give me an answer. It's an exergonic or endergonic? Endergonic, right? They cannot occur spontaneously. Keep in mind, a person is taking a ball back to the cliff, right? Okay. And if I ask why, I hope you are able to tell regarding like what's happening with reference to, the delta G and the other stuff. So now we can see some free energy example of water to ice transformation, and sign of enthalpy and entropy can either be positive or either could be negative. So now look at that delta H will be positive, of course, if you At the heat, it is negative. If the heat is lost during a process or during a reaction, delta S will be positive if the system become more disordered, right? And if negative, if the system become less disordered. So these are the couple of things which we see in this example. It just shows the thermodynamic of the ice water transformation, and you can see the different bunch of temperatures has been shown in the on the left side, and the delta E, delta H, delta S, T delta S, and delta G has been shown. And the decrease in entropy can be seen in the snowflake formation, which I already showed you, that the entropy decrease, disorderness of the system decreases. And now look at this here in this example, right? So -10, you can see this is, first of all, let me give you a little bit. It's an water ice transformation in which 1 mole of water converted to ice at 10 degrees Celsius. 10 -10, 10 degrees Celsius, 0, and -10. So we can see the energy is less in ice than liquid at all these 3 temperature, but at higher temperature, like here you can see that entropy is more negative, right? Look at here, it's more negative, and you can see, because ice is here, you can see highly organized, right? Highly ordered. So that's what I was keep explaining you in couple of previous slide. I hope everything tied up here at this point and making more sense. I know that's when I know it's easy to just go and just deliver those content, but when you have to remember, you need to literally think about it, what's going on, what's the relation between delta H, delta S, and entropy. That's why I try to explain like with couple of other pictures and those models, whatever will help you. Please feel free to use those slides. Okay, so now it's kind of like more chemistry, free energy changes in chemical reactions. So what we are doing. So this is basically a chemical reaction in which show A plus B. Let's say these are your reactant forming C plus D. These are your products, right? And this is shows the reversible reaction. When this product is formed, this again leads to form reactants. So basically, all chemical reaction are theoretically reversible, right? Theoretically, I said, not 100%, right? We know there are some reaction which are irreversible mostly, right? The rate of chemical reactions are proportional to the concentration of reactant and at the equilibrium. So most probably you have done in your chemistry, rate of forward reaction is directly proportional to A plus B. Whatever the reactant you have, amount you have it, that's what you will get the yield of the product, right? And when you remove the proportionality sign, you put an equal sign and replace it with the equilibrium constant. It says K1, and rate of reverse reaction is also directly proportional to the concentration of, these signs are representing the concentration of C and D. And when you remove the proportionality sign, you say, let's say K2. So when you organize in chemistry, rate of forward reaction is equal to rate of reverse reaction. In a reversible reaction, you write down this equation here, you write down this equation on that side, you take the K2 on this side, AB goes on this side, and we replace K1 and K2 with one single, a word which is called equilibrium constant. So all chemical reactions spontaneously proceed towards equilibrium. So that's what we are basically showing in chemistry. And remember, the ratio of this K1 over K2 is termed or is called the equilibrium constant, that is K equilibrium. So please keep in mind. So equilibrium constant basically it predicts the direction of the reaction. Remember I told you when we talking about delta G, it doesn't tell you the direction, right? So it actually predict, I didn't say it exactly, it predicts the direction of reaction. Let's say you put the values, you have the same amount of the reactant and product, you end up with 1 here. You plug in the different numbers and you got equilibrium constant, which is 9. So what does this mean? If you get some value which is greater than 1, it means the reaction will proceed at a greater rate towards the formation of products, which is C and D. If you get a lower value, because these both are greater than 1, right? This one is greater than 1. So if you get a less value, K equilibrium is less than 1, it means the reverse reaction will proceed at a greater rate than the forward reaction. So basically it's just a way of representing and knowing the reaction is proceeding either in the forward direction or either in the backward direction. So that's the K equilibrium actually showed us how we proceed further. Okay, so the greater okay, let's talk about free energy changes in a chemical reaction. So we talk about as long as the total free energy of reactant is greater than the total free energy of product. Remember, keep in mind those examples, the delta G has a negative value and the reaction proceed in the direction of formation of product. Is basically a sum of those two slides, which I already covered. If you have greater delta G, right, the farther the reaction is from equilibrium. So, and the more work that can be performed by the system. Remember this thing in mind. At equilibrium, the change in free energies of product and reactants are equal to 0, which is showing here. Okay, and again, I'll mention as the reaction proceeds, the difference in free energy content between the reactant and product, they decreases and the delta G becomes less negative. So this is a way of showing it's a 1 equilibrium constant, which we showed in the in the previous slide. And if it is okay, so now we talk about free energy changes in chemical reactions. So basically, free energy changes of reaction are actually, we are comparing it under standard conditions. And the standard free energy changes, which is called represented by delta G naught, it's subprime, and they are described for each reaction under specific conditions. So basically, what are these specific conditions? So the temperature is 298 Kelvin, 1 atmospheric pressure, and all the reactant and products, they are present at 1 molar concentration. pH should be 7, except for water, which is present at 55.6 molar, and not hydrogen at 10 to the power minus 7 molar at 25 degrees Celsius. So now we are actually looking the standard conditions, which are not representative of cellular conditions, but they are useful to make comparison. And the most important thing is that we need to always keep this in mind, that the standard condition do not prevail in the cells, and therefore one must be cautious in the use of values for standard free energy differences in calculation of cellular energetics. And if we look at this, standard free changes are related to the equilibrium, and delta G naught is equal to minus RT. It's a log n K equilibrium constant, and this is a natural log. When you convert to log 10, you can write down the equation as minus delta G naught prime equals minus 2.303 RT log equilibrium. You don't need to do this. It's just sort of free energy changes. How you want to write it and represent it in a chemical reaction is being represented here. So R is a gas constant, which is 1.987 calories per mole Kelvin. these are calorie, sorry, that absolute temperature is 298 Kelvin. And if you recall, your log 1 value is always 0. So it's just a way of showing it's represented how you are representing in equations. Okay, so free energy changes in chemical reaction. But you need to concern and remember the most important thing. So reactions having equilibrium constant, which greater than 1. Again, I'm talking about the equilibrium constant, right? It's if it is greater than 1, have negative delta G naught prime values, which indicates that reaction can occur spontaneously under standard condition. And the reactions, if having the K equilibrium constant is less than 1, it means that it has positive delta G naught prime values, and it indicates that reaction cannot occur spontaneously under standard conditions. So now we know that delta G naught prime, it's a fixed value for a given reaction and indicates the direction in which the reaction would proceed what the system at in the standard condition. And again, I mentioned that these standard condition actually they do not exist within our cell. So we cannot, delta G naught prime is a value cannot be used to predict the direction in which a particular reaction is going or proceeding at a given moment within a particular cellular compartment. So is just a way we are showing. And again, this is kind of again reiterating. So negative value means the reaction is again spontaneous and go to the right, all under standard condition. And positive value means that the reaction will proceed to the left. That the reverse reaction is favored, all under standard conditions. So now some important points here, if we can look here. So it shows, this table shows the relationship between delta G naught prime and K equilibrium at 25 degrees Celsius. Again, we are considering all those, standard condition, keeping in our mind. So in this graph, first of all, you can see that the standard free energy changes, they have been plotted versus the equilibrium, this constant here. And what can you see here? So the greater delta G naught negative value, the farther to the right the reaction will proceed before equilibrium is reached. So if you look here at this graph, so greater delta G naught prime and negative values, if you can look these one, right? So the farther to the right, the reaction will proceed until or unless the equilibrium is reached, right here. And if you look delta G naught positive values, which are here, you can see the positive value reaction will proceed to the left, and reverse reaction will proceed. Because again, keep in mind that when we talk about and we even draw this kind of plateau, we know that this is the equilibrium constant here, which I was explaining in the previous slide. And if it is going to this direction, we know if it is less than 1, right, so where it should go, and if it is greater than 1, so where you are predicting that your reaction is going to. So these are some free energy changes in chemical reactions. And now we will see calculation of free energy changes in metabolic reactions. So under standard condition, as I mentioned, that we cannot use these to predict the direction of a reaction proceeding at a given moment of time. So non-standard conditions are basically corrected for the prevailing conditions. So this is an equation here, delta G naught prime plus RT and log N. And by putting this, again, we don't need to concern too much about this. This is kind of the chemistry, which is like mentioned in your book, and I want you to get familiarize yourself with this concentration thing. This is again Uh, uh, Again, kind of repetition that prevailing condition may cause delta G to be negative. Remember this thing, even when delta G naught is positive. Now, making delta G negative may involve a coupling endergonic and exergonic reaction in a sequence, which I'm going to talk in a minute in next slide. And simultaneously coupled reaction have a common intermediate. What does it mean? That is this common intermediate which you see in your reaction. It could like to get rid of the product so quickly because the product is the reactant of the next reaction. Which we will see again in a couple of next slides. And ATP hydrolysis is often coupled to endergonic reaction in cells. Okay, so this is an example of ATP hydrolysis, and you can see here this is an ATP, adenosine triphosphate molecules. It attached with these 3 phosphate group. And if you look here, what is the interesting thing showing here? This is delta G naught prime is showing minus 7.3 kilocalories per mole, right? So it form ADP and PI in organic phosphate, and here it's a small arrow which shows that it can go to the reverse reaction, which is plus 7.3 kilocalorie per mole. So now this make you think or maybe ask the question that ATP hydrolysis, what sort of a reaction it is. So basically looking at this, we will say it's an exergonic reaction. And talking that why it is, so we can say that delta G might be more negative in the cell because ATP is kept at a higher concentration. So we need to keep a couple of these things in mind when we talk about these things. And what else I want to talk about, the greater delta G negative value, the farther to the right the reaction will proceed. So these are a couple of things we need to know. Now, why is ATP hydrolysis is so favorable? This is a very, very important question, and I might, I can ask you this in exam as well. So now I wrote actually the points just to give you kind of an answer. If you can write down these, you will get full points. So ATP hydrolysis is a very favorable, even though it's an exergonic reaction, right? So why is favorable? So it's because of entropy. And why it is, so basically decrease in free energy favors the product formation, and thus it increase in entropy. If we look at the other thing is electrostatic repulsion, and thus electrostatic repulsion is actually creating happening because the adjacent negative charges repel each other. If you look at this structure here, they have a negative charges which is going to repel each other, right? The third important point is resonance stabilization of the product. And basically, I don't know, you know this and you have did this. So basically what happened, this resonance stabilization is a way of describing the delocalized electrons in a molecules. And these molecules are wandering here and there, and because they are not at their specific position, so it's not easy for them to go and make a bond with some other products. So what we see, the resonance stabilization of this ADP and PI is basically is greater than that of ATP, and ATP is sitting here again. I have to go back. So it's favor this hydrolysis because it's in perfect position for it. So that's the other point. And the last one is solvation of ADP, PI, and H positive is higher than ATP. Solvation means kind of, making a solution or something. So it is easier for ADP to lose one of its phosphate group, basically, but it takes large amount of water to force ADP to lose one of its phosphate group. So if so many things are in the favor of or giving an advantage, switch to ATP. So definitely the ATP hydrolysis is always favorable in the cells, and we do need lots of ATP. We know that because our body is actually working, we need lots of ATP to perform our body task, right? Okay, so now we will talk about a little bit more about the coupling endergonic and exergonic reaction. And here we can see role of for ATP hydrolysis in the cell. So why we need to hydrolyze this ATP hydrolysis in cells is required so often and so much. So look at the example. There are 5 examples given. I'll go and explain one by one. So first, it separate charge across a membrane. And you can see very, very clearly that it can, this membrane is shown here, right? And you can see it has positive, negative charges are here, and the ADP comes here, and we can see that it aligned basically those charges here, right? So that's the important thing. And the second point is it's concentrate a particular solute within the cell. So if you look at this, it's the ion transporting protein, and basically it's helping those potassium ions to concentrate inside this within the cell. The third one is it drives the chemical reaction, and how it drives the chemical reaction. We know this is amino acid, right? Glutamic acid. The ammonia is in the body, in the cell, it's there. And basically there is an enzyme which helps to make a glutamine. And you can see is delta G is plus 3.4. Right, so it will actually what happened, this is an intermediate product, and this enzyme will come here, and it will with the help of, you can see ATP, that second, this is just a basic way of showing that is a plus 3.4 delta G values has been shown. So in the first reaction, glutamic acid needs this ATP to make glutamyl phosphate and ADP. And now in the second reaction, because this is not very clear, this glutamyl phosphate basically combined with this ammonia. That's what's shown in your book, and this form the glutamine phosphate and release the PI, and this ADP again, of course, going for recycling. So again, this is minus 3.9 kilocalories per mole. So you can see how this ATP play important role in the cell, to perform some sort of a chemical reaction. Now the fourth example, movement of filament in a muscle cell. When we also talk about this, that we have filaments in our body, which helps us to slide the filaments on the top of each other and help the muscles move. But basically they require ATP, and they cannot work and form those sort of bridges, and you cannot contract your muscles. So basically ATP helps that. To facilitate this process. And the last one is changes in the properties of protein added and also helps to perform their function, which they are required to. So this is a protein, it's required, with the help of ATP, the phosphate group attached, when it breaks down, phosphate group attached, and it sort of changes conformation shape, and now you can say serve as a binding site for other proteins to perform other functions, which is quite common in cells, and we see it most of the time. Now we are talking about equilibrium. Let's see, if a complete equilibrium attained in the cell, it means death of the cell. And how it happens, so basically let me just move this. Okay. Equilibrium versus steady state metabolism. And here what I am going to show you that cellular metabolism is non-equilibrium metabolism, meaning that it never comes to the equilibrium. Once it comes to equilibrium, it means death, which I showed it here. So cells are open thermodynamic system. We talk about that, and this is an example. You can see that cellular metabolism exists in a steady state. And here you can see that the concentration of the reactants are basically, and products remain constant, but not at the equilibrium. So look at this amoeba. It's shown here. So it's releasing carbon dioxide, and it's like, you know, the things are coming inside. So in a steady state, ATP is low, ATP is high, just like we saw in our body. But what happened when the equilibrium is achieved, then ADP gets high and then ATP gets low, and you can see there is no movement of the molecules is happening, right? So new substrates and end products basically are removed here, and it says maintaining a steady state require a constant input of energy, whereas maintaining equilibrium, it doesn't. And we can see clearly here in this picture, right? Okay, now we are almost coming to our laws of thermodynamic, and this is a third law, which states entropy is 0 at 0 Kelvin and it's minus 273 degrees Celsius. This is just the simple third law, while the 0th law is basically talking about that this A and C, let's say they are in thermal equilibrium, and they are brought into contact, there would be no heat transfer. So basically A in equilibrium with B, and B is in equilibrium with C. So it's kind of a transitive phase, right? So that's what is that one. And now this is kind of, it's a summation of your laws of thermodynamics, right? So it's a first law, second law, and third law. It's been shown here. So that I'm not going to repeat it. You can just see, have a look. It's just a summary. And I have almost done. I'll just take 10 more minutes for this because that's our lecture time. And then I will cover the rest of in our Thursday lectures, which I'm going to post it as separate. So just to give you a little bit background, what are enzymes, you most probably know it. And the other thing is that we are doing a lab on that as well. The lab 5 is all about enzyme. So basically, if we talk of our enzymes, it's started you know, like there is a history basically. So there is a chemist who actually prepared a yeast juice, and what he did, he actually took you know, the grind. He took the because the yeast juice was like it'd been known, right, for a while. So he thought, okay, he can grinding yeast with the sand, and what he said, he said, okay, I have a yeast juice. Now he wants to preserve the extracted that yeast juice with some sort of an antiseptic, and so he decided to, okay, I'm going to add sugar molecules into it, because previously in that era, you know, in 1800s, it's been known that if you want to save your food, you know, you put sugar or put so much salt, you know, that's where the pickles and those kinds, jam and jellies concepts come in, to save it. And because they don't have like the food like us, just go to, you know, grocery stores and buy it. So they have to preserve the extra food because to avoid themselves to go and hunt in winter. So basically the short story is that what he found when he put sugar, he found in an interesting thing. So basically what he did, he when he grinded the yeast with the sand, you know, so he filtered it in the filter paper, and he had that juice, and he put the sugar to preserve it. And now what happened, after several days, he saw, oh, there is like some sort of a, you know, gas is coming out from that. So that's give an idea that you do not require the presence of intact cells for to for this yeast, and he gave them the name enzymes. So basically, it's a Greek word of yeast. So that's what it comes in. Now, the study of enzymes is called enzymology. It's a mistake here. It's a is. Now, over 10,000 of enzyme in every single kingdom in the phylogenetic tree of life have been discovered so far. Enzymes are known to catalyze more than 5,000 biochemical reaction types, and over 10,000 different enzymes have been identified so far. So these are some of the facts about enzymes. The most important thing is that enzymes specificity come from their unique three-dimensional structure. That is the most important point, and you need to remember if they lose their three-dimensional structure, which we saw in our chapter 1. I'm going to keep reference the stuff we already covered. Remember mercaptan and ethanol stuff, which you use it to fold the protein, unfold proteins, right? So if they lose it, the structure is lost. And the enzymes are mostly made up of protein. They cannot perform their function. So enzymes are how we name enzymes. So enzymes are named by adding suffix, which is "-ase" to the name of the substrate they belongs to or they are acting on, or basically the type of reaction they are catalyzing. So now enzymes as biological catalysts. So I will, after this, I will stop. So enzymes act as catalysts. And if I say, can you give me some enzymes? So there are lots of enzymes around us, like ureases, phosphatases, right? Decarboxylases, dehydrogenases. So basically, enzymes, they act as catalysts. What are catalysts? You know it most probably. They do not take part directly into the reaction. Rather, they like to increase or they speed up the chemical reaction. And many thermodynamically feasible reaction in a cell that could occur do not proceed at any appreciable rate. That is ATP hydrolysis. Enzymes are almost always protein. I wrote the word almost, not all, remember, because if you remember your first chapter, I also talk about, and we also discuss more that RNA can act as a catalyst, right? And they are called ribozyme, RNA polymerases there, right? So there are which synthesize RNA from a DNA strand in transcription, right? So they catalyze biochemical reaction. And enzyme may be conjugated with non-protein components, which are cofactor to perform their proper functioning, and either inorganic enzyme conjugate either metals like iron would be associated with them to perform their function, or organic enzyme conjugates are called coenzyme. Okay, I will most probably go over these cofactor and coenzyme concepts. I'll make a slide and kind of make an interactive tutorial for this one, this coming class. So I'll go more and you will learn more about these kind of that stuff. But you will get marks for that. Okay, so I'll stop and I will put the rest in as a Thursday lecture recording.